Today we're going to be talking about blood composition, hemostasis, things like that. So we're going to get started. Let's see. All right. Like I mentioned, we're going to be talking about blood composition, how RBCs, red blood cells, are removed from circulation, and the mechanisms of hemostasis. Hemostasis means the cessation of bleeding, or how we form clots, so that we don't bleed out. Okay, so blood composition. Most of our blood is made up of plasma. Okay, plasma is 55% of our blood, and 92% of plasma is made up of water. The other small percentage of plasma is made up of albumin, globulins, and fibrinogen. These are all helpful in different things. This is especially helpful in our immune system, globulins. Albumin is important in BCOP, and also in helping us take care of when we're breaking down our red blood cells. We'll talk about that in a little bit. And then fibrinogen is really important in clotting. Okay, so this is kind of just a picture. So we have plasma, 55%, and then the other 45% is made up of a buffy coat, which is made up of white blood cells and platelets. And then the rest is made up of our red blood cells. Okay. So that's kind of the blood composition. We're going to move on. There's lots of little proteins in our plasma. Here's kind of a quick little diagram. So most of it is albumin right here. The other largest one is this. They're called gamma globulins. And they're, like I said, they're important in our immune system. They help us form antibodies. And then we also have this alpha and beta band. A little bit of this is fibrinogen. This one's just other clotting factors. So just a little look at that. So now we're going to jump into how our red blood cells are removed from circulation. So hemoglobin, red blood cells, they are broken down every 120 days by macrophages, just because we're constantly crushing them. Every time we bend an arm, or like you're sitting on your knees, anything like that, we're bending them, we're distorting them, and so they break down every 120 days, and they need to be recycled, right? So what macrophages do is they envelop these red blood cells, the red blood cells. And what they do is they break it into amino acids and a porphyrin ring. Okay. Hemoglobin is super important for the transport of oxygen to our tissues, right? So when the red blood cells get into the lungs, they get oxygenated. So they put an oxygen on there, and then they transport that oxygen to someplace in the body, and then they're deoxygenated, and so then they go back to the lungs to be reoxygenated. Follows that whole thing, right? So then when the macrophage breaks down the amino acids, those amino acids are immediately recycled and used in whatever form they may be needed. The amino acids aren't going to be taken away. They can be recycled quite easily. But the porphyrin ring, however, is not so easily recycled. So then the porphyrin ring is broken into an iron and biliverdin. I don't know the best way to draw this all out for you guys to understand what it kind of looks like. So I'm just going to do a really kind of a crappy job of drawing it, but I hope you'll understand. So hemoglobin is a bunch of amino acids, usually 4 actually. All right, let me add another. Okay, and then each of them have this porphyrin ring in the middle. And that's what helps us bind the oxygen. Okay, so these amino acids here, like I said, they're reused quite easily, but it's these porphyrin rings that need to be excreted, released from the system because they're not recycled. And then these porphyrin rings are made up of iron. They have an iron group in the middle of each of these. And then when that iron is removed, then what's left is what's called biliverdin when it's still inside the macrophage. Okay? So what happens to the iron once it's taken out of the porphyrin ring is it attaches to or is connected to a protein called transferrin, and then that transferrin repurposes that iron by taking it to red bone marrow for reuse, right, to create a new porphyrin ring. Whereas biliverdin, the leftover piece of the porphyrin ring, is kicked out of the macrophage, and then that is converted into bilirubin. And then what happens is albumin picks up that bilirubin in the blood and it takes it to the liver. Okay? So once bilirubin makes it to the liver, it's conjugated by adding these glucuronic acids. So bilirubin makes it in to the liver, glucuronic acid is added to the bilirubin, and it's now called conjugated bilirubin. Then conjugated bilirubin is secreted into the bile, or into the bile duct, which is then dropped into the intestines. Okay? Once in the intestines, glucuronic acid is removed by bacteria. So then it's no longer conjugated bilirubin, but is now called urobilinogen. So again, once the glucuronic acids are taken off of the conjugated bilirubin, it becomes urobilinogen. So then there's a couple of different paths that urobilinogen could follow. It can either be reabsorbed into the bloodstream, so leave the gut, leave the intestines, enter the bloodstream again, and then it can either go back into the liver and be reconjugated and do the whole cycle all over again, which is called the enterohepatic urobilinogen cycle, or it can go into the kidney and be converted into urobilin and then be excreted as urine. What's interesting is urobilin is yellow, has a characteristic color of yellow, so that is why our urine is yellow, is part of the reason. The other thing, if it doesn't leave the intestine, if it's not able to get out of the intestine, is it stays in the intestine, and then it gets converted by bacteria even further from urobilinogen into stercobilin, which is dark brown in color, which is why our feces is brown. So this is kind of a nice word chart, right? But I always like pictures, so I've kind of drawn out what it kind of looks like here. So we've got bilirubin, albumin's brought it to the liver, so bilirubin makes it into the liver here. It gets conjugated by these glucuronic acids, okay? And then it's now taken through the bile duct, put into the intestine. So we have this conjugated bilirubin here. Bacteria eats away at the glucuronic acids. It now becomes urobilinogen. Urobilinogen is able to cross these walls, and it can either re-enter the bloodstream or it can keep going down through here. If it keeps going through, it becomes stercobilin, which is then excreted. Again, dark brown in color. And then, or if it decides it can go back up it wants to go back into the bloodstream, it can either go right back into the liver where it's reconjugated, or it can go into the kidney, become urobilin, and be excreted as urine. I don't know, whatever is most helpful to you guys. Take a look. It doesn't matter to me. I prefer to see a picture. A picture seems to stick in my mind best, but up to you guys. Now we're going to move on to the mechanisms of hemostasis, cessation of bleeding, how we stop bleeding when something goes wrong, right? So the first thing that happens is there's a vasospasm or a vasoconstriction or whatever you want to call it, I guess. Yeah. So what happens is, there's pain. There's something that happens that ruptures a wall, right? That hurts. And so that causes a stimulation of smooth muscle to constrict around the blood vessels, which decreases the amount of blood flow through that blood vessel. Now, this is only a temporary change. It's not a long-standing change until the cell wall or the trying to think. I'm just having until the problem is fixed. It's only a temporary fix here. What is really important is the platelet plug and coagulation. These two things are what are going to help us stop the bleeding long term. Okay, so we're gonna kind of dive deep into platelet plug and coagulation. So first, what happens in platelet plug? Like I said, I like pictures, but I am gonna just go through this this word chart first. So this VWF, it's usually von Willebrand factor, but we're just going to call it VWF. That is secreted when there's that pain, when there's that damage to a wall. Okay, so it's secreted, and then what VWF does is it binds to platelets, and then it binds platelets to collagen in the cell wall. So if I'm going to come up here and just kind of show you. So I have this cell wall, and then here's some damage, right? And then the cell wall continues on back over here, okay? But in this damage site, collagen is exposed. So what happens is if this is von Willebrand, VWF attached to a platelet, and it comes up here, then it'll attach to collagen. So now we have VWF attached to both collagen and a platelet. And when that happens, that platelet is activated. So we went from this to this. So coming down here, once that platelet is bound to collagen, it becomes activated, okay? And what happens when a platelet is activated is it changes shape, which is why it went from a circle to a star shape in my drawing up there. It also has... platelets have these granules here, alpha granules and dense granules. Alpha granules release more VWF, fibrinogen, and what's called platelet something growth factor. I gotta find it. Hold on. I don't want to lead you astray here. Oh, platelet-derived growth factor. So, alpha granules release more VWF, fibrinogen, and platelet-derived growth factor, PDGF. Dense granules release several things, but the most important thing is ADP. Okay? And then also, when a platelet is activated, we see TXA2, which is thromboxane, is released from the membrane of an activated platelet. Okay, TXA2 and ADP are important in activating other platelets. So when they're excreted or released or whatever you want to say, they go and activate more platelets. Okay, and then another thing is VWF and fibrinogen receptors appear on activated platelets. So when they're not activated, we don't see these receptors on there necessarily. We see some VWF, obviously, because they have to attach to something, but as they become activated, we see more of these. Oh, and then, like I said earlier, TXA2 and ADP activate more platelets. So they're bringing more to the cause here. And then what happens is fibrinogen is really important in clotting. As we mentioned up above, fibrinogen binds to high fibrinogen receptors on activated platelets, and they link together. and they form this bridge here. So like I have written here, activated platelets stick together because of fibrinogen receptors. Fibrinogens form bridges and they cause them to stick. And so they kind of bridge this whole gap here. This fibrinogen keeps them all stuck together so that they don't lose each other. And that helps bridge the gap. Okay, so if VWF is all these little green guys here, they're going to attach to a platelet, and then they'll come up here attached to the collagen, and that will activate a platelet. The platelet will then has these alpha granules and dense granules. Dense granules release ADP, and then just the membrane itself releases TXA2, thromboxane, and they come and they activate more platelets to the cause of fixing this problem area here. Okay, and then as these all kind of start coagulating or coming together, right, getting more and more to cover up this area, fibrinogen is attaching to these fibrinogen receptors, and then we're creating these little bridges that help us stick together. And that's a really important part of the platelet plug. Okay, and then at the same time that this is going on, we jump down here to the coagulation cascade. Okay, so these are happening simultaneously. It's not like the coagulation cascade waits until we have a nice full-on platelet plug. They're happening simultaneously. Okay, so now we're going to move on to the coagulation cascade. There's really not any super easy way of teaching this. It's just kind of brute force memorization. Okay, so what happens is we have this factor called factor 12, or the Hageman factor. And what happens is when there's this opened up gap here, this site of injury, it opens up collagen, right? And collagen has a bunch of negative, it's negatively charged. And so when this factor 12 interacts with those negative charges, it becomes factor 12A. Okay? And then factor 11 comes in, and it interacts with 12A, and it becomes 11A. And then factor 9 comes in, interacts with, or it's not actually a factor. What it needs is the help of factor 8 to affect factor 10. Okay, but anyways, so 12 to 12A, 11 to 11A, and then 9 to 9A plus 8. And then 10 will come and interact with these two here, and it'll become 10A plus 5. And then we have factor 2 interacting with that, becoming 2A. And then we have factor 1 coming and interacting with 2A and becoming 1A plus factor 13. Okay? Now, this is interesting because we have what's called the intrinsic pathway. It's more like right here. This is where it kind of ends. And then it becomes common. Now, what we mean by common is we have this intrinsic pathway, and then we have an extrinsic pathway over here. At this point, right here, things become common between the two pathways, both intrinsic and extrinsic. Okay? So at this point, 10A plus 5 is what's happening. Okay, and then everything beyond that is the same. So the extrinsic pathway is we have this factor 7, and it's coming up into here, and we have these cells up here that have factor 3, which is called the tissue factor. Okay, and so there's also some here in the collagen, but this factor 7 will interact with factor 3 and become 7A. And when it becomes 7A, then we have factor 10 come interact with 7A and become 10A plus 5. Okay, and then at that point, we become the common pathway, or we turn into the common pathway. So I tried to draw it out maybe just a little different so that it maybe makes a little more sense. So again, we have the intrinsic is 12 to 12A interacting with some negative charge here. Okay. 12 to 12A, and then 11 to 11A, and then 9 to 9A, plus 8. Okay, so that's kind of the start. That's the intrinsic pathway. The extrinsic is 7 plus 3 tissue factor goes to 7A, and then both of these interact with 10, or 10 interacts with both of these. So then 10 interacts with 9A plus 8, or 7A, and becomes 10A plus 5. And then 2 interacts with 10A plus 5 and becomes 2A. And then factor 1 interacts with 2A and becomes 1A plus 13. So I hope that maybe made a little more sense. Drawing it out this way helped me understand it better, both the intrinsic and the extrinsic. I think of intrinsic as happening at the site of the problem, right? Like it's in the thick of things here, right? This is the problem site. So it's inside the problem, and then extrinsic is external. It's happening farther down. There's no site of injury here, but it is interacting with this tissue factor, and that is coming over and helping out with the clotting cascade, helping us all clot everything up. This was something we talked about in class. I'm not sure exactly if they want me to teach this or not, but the most important factors that they wanted us to know were these guys right here. So factor 12 is the Hageman factor. Factor 3 is tissue factor. Factor 2 is prothrombin. Factor 2A is thrombin. Factor 1 is fibrinogen. And factor 1A is fibrin. Okay. Now, how do we break all this stuff up? Once the area is healed, and we need to get rid of the clot, or the bridges that were made in the plug, the platelet plug, and everything, is we have what's called this TPA. It's the tissue plasminogen activator. So we have plasminogen. TPA comes in and activates it. Plasminogen becomes plasmin. Okay, and then once plasmin is made or activated, then it basically calls on fibrin to auto-destruct, basically. So plasmin doesn't really do anything. It just, fibrin recognizes, oh, hey, there's plasmin now, so I'm going to start cutting up all my stuff. So again, here's kind of a word chart. This might make a little more sense, but the endothelial cells produce this TPA, tissue plasminogen activator. When fibrin is present, TPA accelerates enzymatic function to convert plasminogen to plasmin, which then initiates clot dissolution. So it initiates the clot to break up. By activating TPA, fibrin initiates its own degradation. Kind of interesting. But so that's pretty much everything we had to talk about today, guys. I hope that all made sense. Thanks for watching.